right, in chapter 16, we're going to switch gears kind of all together. So we're going to talk about microbial life, some of the real small things. So we have two major sections for chapter 16. We have prokaryotes and protists. Prokaryotes is where we'll spend our time right now. And hopefully this will be a good review for you because we've covered prokaryotes in the past, but now we need to add a little bit more to it. So at least you can kind of rest assured, because there's a lot of information here, but you can rest assured that some of it's stuff you've seen before. We're going to add to that. So our essential question is, what are prokaryotes? And then we're also going to attempt towards the end of this video to figure out how they're classified. So we got to do both. We've got to define prokaryotes, which, oh wow, class. I forgot what I was doing. I was talking so much, I forgot what I was writing. There we go. Okay, so we're going to look at prokaryotes, and then we're also going to talk about how they're classified. Okay, so we already sort of know what they are, but we need a little bit more detail to them, and we need some different ways to describe them and classify them. So hopefully this is a review for you, is that prokaryotic cells are smaller than eukaryotic cells. So we have the general range is about 1 to 5 micrometers or micrometers is better in diameter. So about 1 to 5. Eukaryotes, on the other hand, are 10 to 100. So they can be anywhere between 2 and 20 times or even 100 times larger than some of our prokaryotes. So eukaryotes are much larger. However, the collective biomass of prokaryotes is at least 10 times that of all eukaryotes. So it just says that there's many, many more prokaryotes than there are eukaryotes. So many more of them. And we mean like lots more. Because think of how many prokaryotes it would make to make up a eu. Like, we're already made of hundreds of thousands of cells. And then we've got smaller cells just to make up us as one organism. It takes that many organisms. So there's so many prokaryotes. Just so much more. And biomass, by the way, is like the mass of that particular biology. So the mass of that particular living organism. So these living organisms far outweigh us. Alright, so here's some little guys just on whatever this is, probably like the tip of a pen. Just some prokaryotes there for us to look at. So where do prokaryotes live? If you remember, because I haven't really mentioned at this point, remember pro is sort of before, and then kary means kernel or nucleus. So these guys are before the nucleus, so no nucleus. In case we brain dumped that one, we gotta kind of pull that one back out. Take the cobwebs off of that one. So we're talking about no nucleus. These guys, however, can live in all kinds of habitats. So they can live in places that are too cold, too hot, too salty, too acidic, too alkaline, which kind of means too basic, for eukaryotes to survive. So they live in these diverse habitats, which you can see why they have more biomass. So there's a lot more of them in all these places that we can't go. Some bacteria are pathogens, which means that they cause disease. We've all experienced one that caused disease. Okay, strep throat, tonsillitis, things like that, even ear infections and stuff like that can all be caused by these pathogens. But most bacteria in our bodies are benign or beneficial. So they're either don't do anything to us or they're good for us. All right, and so that's what we look at. Like all the stuff you think probiotics is a good one that's beneficial. So our microbiota, all right, so it's the microbiota would be the community, let me see if I can write here, the community of microorganisms that live in or on our bodies. Okay, and so that's going to be all those things that are in your gut. When they say take these and you get probiotics, it just means that you're adding good, happy bacteria to your digestive system. They live all down in your stomach. Maybe stomach's a little tougher to live in. Like small intestines, large intestines for sure. On your skin, there's a whole bunch there. That's fine. So we just have lots of little microorganisms that live in or on our body. Each of us harbors several hundred different species and genetic strains of prokaryotes, including a few whose positive effects are well studied. So some examples of these positive or beneficial ones would be that intestinal bacteria that we're talking about. They actually give us essential vitamins and enable us to extract nutrition from food molecules that we otherwise can't digest. So we need them because they can give us extra vitamins. All right. Many of the bacteria that live on our skin actually perform housekeeping functions, such as decomposing dead cells. So they're little housekeepers, like, oh, this one's, let me just boot this out of the way because it's not really living well anymore. It's not being helpful. So we've got some cleaners. We've got some that help us get more nutrients out of our bodies, which is amazing. Other prokaryotes guard against pathogenic intruders. So that's the whole point, is that they're little guards. If something else tries to get in close to your body, it's like, no, this is my space. Fight, fight, fight. And then you don't have it. Maybe not quite that dramatic, but I like to imagine it. Little prokaryotes fighting each other. That'd be cute. Okay, so prokaryotes in the soil help to decompose dead organisms. So they're used for decomposition. And that's how we're able to get rid of things and help kind of recycle organic waste materials, which are vital chemical. Let's see if I can get words out tonight. Vital chemical elements for our environment. Let's see. Phone went off. All right. I'm distracted. Like you guys never get distracted, right? All right. Let me get back on it. If prokaryotes were to disappear, the chemical cycles that sustain life would halt, and all forms of eukaryotic life would be doomed. Okay. Oh my goodness, I'm trying to be funny and it's not working out. Anyways, it's sadness, and we would be doomed. There you go. All right, just to help with some classification. So again, we've got to classify two different ways. So we have two very different types of prokaryotes, which I think we covered a little bit in the last lecture. So we get to go in a bit more detail. So we've got archaea and bacteria. Okay, those two domains. So this is before we ever even get to kingdom. Well, I meant to explain that further. We kind of will, because we're going to go into more classifications at the end. But these guys tend to be more of the extremophiles. They live in extreme environments. That's their habitat. And then bacteria are just kind of like all the other guys. Sort of, we'll put normal bacteria. All the ones that you would think of. So kind of keep those in mind. The archaea seem to be extreme. Archaea extreme. All right. So within these, we have some other classifications as well. Some things that we need to know as far as shape goes. So we've got some common cell shapes. So cocci is a spherical eukaryotic cell. They sometimes occur in chains. So a chain is strepto. Cocci is circular, so if this one were in a chain, it would just be sort of a circular bacteria that's in a chain, just like that. Okay. Bacilli, so you've heard of like lactobacillus, that means it's a rod-shaped prokaryote. So they can be kind of thread-like or filamentous. This is actually what we think of with most of them, sort of like E. coli is sort of like a rod shape or a pill shape. We also have some other ones that are kind of a corkscrew. So if they're short and rigid and they're spirals, then we call them spirilla. So kind of like, however, if they're longer and more flexible, we call them spirochetes. So these are long, loopy things. So the three basic shapes that we're going to work with that are really important for this are cocci, which are going to be round, bacillus, which is rod-shaped, and then spirals, which we can go with spirals. There's spirilla and spirochetes, which we'll see a little bit later on. Okay, so another picture, here's cocci, the round. I always think the letters in cocci are so round, like you get kind of the roundish letters. It's telling you it's a round one. Bacilli, you've got the long L's, which give you the rod shape. Okay, and then spirochete, it's a spiral. Yeah, so it's nice to remember. Okay, some of the things we can go by. So nearly all prokaryotes have a cell wall. So this is one of their things, is they have a cell wall. And they can provide some physical protection, and they can help keep it from bursting in a hypotonic environment. Oh, we have to remember that. So in case it encounters something that has low solutes, you remember that? Low solutes, like pure water and things like that, it can prevent it from bursting. So there's a way to check this, is that we actually have a Gram stain, which stains the cell walls of bacteria. And so if it stains positively, so it turns, I think, like a purplish color. If I remember, it's like blue or purple. Like, so if it turns blue or purple, it turns a color, it's Gram positive. And so these are simpler cell walls that contain peptidoglycan. If it's Gram negative, so it doesn't turn the purple or the blue color, it has less peptidoglycan. And these bacteria are more complex and more likely to cause disease. So Gram positives are kind of your normal, your good ones, and then your Gram negatives are usually, and not always, but usually your not so good ones. All right, let's see. So something you can sort of see kind of in the background, they're a little bit of the pinkish, so those are Gram negatives, and the purple ones over here that have kind of a deeper color are Gram positive. So that's how you sort of tell the difference. All right, this would be a good time at the 11-minute mark that if you need to take a break, you know, wherever you're at, you can take a mental break, even just for a couple minutes, even standing up. So there's some good scientific evidence that says that if you stand up and move around for just a second, it helps to get some of the stuff going to your brain so you can think better. So that might be a recommendation as you go along, is that if you're at the 10-minute mark, even if you're wanting to plow through it, like just pause the video, take a break, stand up, shake it out. You can do the sprinter shakes. If you don't know what I mean by the sprinter shakes, just ask me. But you do those, shake your legs out, sit back down, and it should process better. All right, so we'll move forward here. So the cell wall of a lot of prokaryotes is actually covered with a capsule as well. And so that's a sticky layer of polysaccharides. Oh my God, old vocab. Remember polysaccharides? Poly is many. Saccharides, we're talking about sugars. These are carbohydrates. So the capsule often is made of long sort of sugars or proteins. So the capsule actually enables the prokaryotes to adhere to their substrates or to other individuals in the colony, and it shields pathogenic prokaryotes from attacks by the host immune system. So there's sort of some functional things that we can look at. I don't think I ask a whole lot about it, but it is helpful to know that there is a capsule sort of outside of the cell wall that helps provide some extra protection. Okay. Oh my God, I can't. I'm like trying to put all these words together today. So it's extra protection or it's a sticky factor, and that's all you'd have to write. Again, efficiency in how we write it. So let's say this is a tonsillitis of sorts, is that we've got a tonsil cell and this particular bacterium is attaching. So there would be kind of a cell wall here, and then around it is we have this capsule that allows for sticking and allows for some protection. So prokaryote population growth occurs by binary fission. I feel like I've asked you guys that before, so that should not be new information. So population growth is by binary fission. And population growth is literally just how it sounds. The population grows in size. Okay, so it can rapidly produce a new generation within hours. Within hours, a new population. And it can generate a great deal of genetic variation. So even though it looks sort of like, well, it's just making exact copies, there is, with more copies and more things going on, there can be room for small mutations. So these spontaneous mutations, every time that it has to replicate, there's the chance that we can alter the genetics, which gives us genetic variation and increases the likelihood that some members of the population can survive changes in the environment. Which again is how we sort of end up with something like MRSA. Okay, so the genome of a prokaryote typically has about 1/1000th, and like 1/1000th as much DNA as the eukaryotic genome. It's usually in one long circular chromosome packaged within to a distinct region of the cell that we call like the nucleoid space. Okay? Many prokaryotes also have an additional circular piece of DNA that we call plasmids, which replicate independently of the chromosome. We talked about plasmids, I think, if we did DNA technology. I might be thinking of the college class, so you got to tell me if we did or didn't. But plasmids are often used for DNA technology stuff. All right, so you have like a chromosome, you have this kind of big circular chromosome, but out to the side you'll have these extra little pieces that are circular pieces of DNA. We call those plasmids. Okay. Good for DNA technology, real easy to mess with. We haven't really talked about it because we've had so many times that we've been away from each other, but there's an interesting thing. So if you look up way back in the day, they did this thing with, oh my god, no, it escaped my brain. Did you hate when that happens? Something just escapes your brain. So there was a thing where there were anthrax. There we go. They were sending envelopes of anthrax to various like political figures. It was a really like kind of biohazard, biocrime, you know, type situation. And that's because, so what happened is that prokaryotes of specialized cells end up in endospores. So they were sending endospores, so these dormant kind of prokaryotes that sort of are dormant in harsh conditions. So they'd put this anthrax into this kind of endospore in this dormant phase. And then it could survive extreme heat, cold, unfortunately, and kind of scarily, like the UPS. Okay? But when the endospore receives environmental cues that the conditions have improved, like the anthrax getting kind of inhaled into somebody's kind of nose hairs and things like that, it would absorb water and resume growth and then cause a serious infection. Like, that was a bad one. But that's how they were able to send it, is with endospores. Okay. So here's a bacterium, and these are sort of their little dormant versions, where they're not metabolizing, they're not really doing anything, they're just waiting for conditions to be right. Okay, you can think sort of like the lysogenic cycle of viruses, only endospores are with this actual living virtual organism. All right. Another thing we'll talk about is that prokaryotes exhibit a whole lot of nutritional diversity, a lot more than we do as eukaryotes. So that allows them to inhabit almost every nook and cranny of Earth. So we have two energy sources that are used. We've got phototrophs and chemotrophs. Again, this should not be new for you guys. So photo is light, chemo is chemicals, and troph is still nourishment. Okay, so these guys get nourishment from light, these guys get nourishment from chemicals. That's it. Okay, same thing we've seen before. So we're just revisiting some kind of old terms or talking through some. We also have two sources of how carbon is used. Okay, so you can think like CO2 type stuff and that kind of thing, or consuming of sugars in that form. But we've got autotrophs, so self-nourishers, and then we've got heterotrophs, which have different nourishments, and so they obtain their carbon from different sources. Okay, so just sort of the way to think about that. Again, I'm more happy if you sort of know those terms, because it gets complicated when you start putting them together. Because here's how we could look at it. We have some that self-nourish using light. So they harness sunlight from energy and use CO2 for carbon, just like plants. We've got some that get different nourishment through light, so they obtain energy from sunlight, but get their carbon atoms from other organic sources. So instead of getting CO2, they're going to get other organic sources and then use light to put it together. I know, pretty crazy. And then we have some that self-nourish using chemicals. So they harness energy from inorganic chemicals and then use CO2, right? And then we have some that have different nourishments using chemicals, and so they acquire energy and carbon from organic molecules. Again, there's a lot happening here. I will not ask you to know all that because there's a lot going on. But the idea is that you can still break the words down to still get an idea of what's happening. I just won't be super specific about it. All right, here's a couple of them, like a photoautotroph. This you could do, which is sort of like plants will have oscillatoria. We have chemoautotrophs, so some bacteria can sort of eat through a rock. So it's kind of an interesting one, but that would be a chemoautotroph. We've got a photoheterotroph. Rhodopseudomonas is one of those, and then actually a type of salmonella is a chemoheterotroph. So we'll talk through some of these. They get weird at that level. I know, yeah. One last thing I want to talk about before we go into a quick thing on some different categories, which we'll discuss, I promise. But one last thing is to talk about biofilms. So these are highly organized colonies that attach to surfaces. So for the most part, prokaryotes are independent. They're single-celled, they're not really colonial, they're not really social species, it's not their thing. But they can organize into colony on surfaces. And you've probably seen biofilms before, just like a thin film over like water. So they consist of one or several species of prokaryote. They can also have some protists and some fungi, which we'll get to soon. Biofilms can form on most any support, including rock, soil, organic material, surface of stagnant water. That's where I think we've seen it maybe the most, is on stagnant water. Biofilms communicate by chemical signals, they coordinate division of labor, they collectively defend against invaders, and there's channels in the biofilm that actually can allow nutrients to reach the middle, waste to leave. The whole environment kind of develops within it. It's pretty gnarly. But there is a whole purpose to this stuff, and you can see it kind of lining a tube. It looks gnarly, but it's actually really useful for cleanup for contaminants in the environment because they really have that great nutritional diversity. They're really adaptable, and those biofilms can really sort of work together. So really, as far as cleaning up contamination, prokaryotes have been our kind of go-to for that. So we can help kind of fix some of those parts of the environment. If I remember correctly, too, there might even be a type of bacteria that can eat plastics. We'll have to, I'm going to write it on here so you guys can help remind me. Plastic eating bacteria? If there is, it's like, that's like a win for mankind, you know? It's like, oh, so many plastics we've used and screwed it. I'm just as guilty, you know, we messed the whole earth up with that. Another term here is bioremediation. So it's the use of organisms to remove pollutants. So remediate, to go back and help and, you know, get back to status quo. So we can try to do that with life for soil, air, and water. Okay. Real quick, decomposers are mainstays of sewage treatment facilities. This is how we're actually to get raw sewage first passed through screens and shredders. Solid matter settles out of the liquid waste, forming sludge. Sludge is gradually added to a culture of anaerobic prokaryotes, including bacteria and archaea, and the microbes decompose the organic matter so it can be placed in a landfill or used as fertilizer. So when we have sewage treatment, we're using these prokaryotes to help us do it. Liquid waste would be treated separately, and we actually kind of can spray some rocks. We use some biofilms, and we'll drain in order to release it into the ocean. So, interesting stuff. We got a whole bunch there. There's actually a water treatment facility in Humble, I think off the Beltway, that's an interesting one to kind of look through this stuff further. All right, it's a good time to take a break if you need to. Again, these are all useful tools, but I want to get to the last part here. Okay. Bioremediation for all these dang oil spills. Okay. Take a break if you need to, and this will sort of be the last thing that we'll wrap up here. So, new studies of representative genomes of prokaryotes and eukaryotes strongly support the three-domain view of life. So we've got eukaryotes and eukarya, and prokaryotes have bacteria and archaea. And so we need to classify these just a little bit further. But we've been able to, through some RNA sequences, RNA polymerase, some introns, some of those peptidoglycan situations, and histones. Lots of things we use to compare these. here's kind of what I was getting at a little bit earlier, so it's a little, wraps that back into it. But the archaeal inhabitants of extreme environments. So these are what we call extremophiles. Philes mean they love the lovers of. Okay, so lovers of extreme. So they have unusual proteins and other molecule adaptations that enable them to metabolize and reproduce efficiently or effectively. So we do have some extreme halophiles. Halo means salt. So we're talking about salt lovers, extreme salt lovers, so they're in very salty places. I feel like there's probably a joke on that one. Have at it on your own time. They're salty. All right. Thermophiles, and so they love heat, so they thrive in hot waters, geysers, even acid pools. So we've got the extremophiles there. All right, some other ones that we have that are extremes are these methanogens. Okay, so methanogens live in anaerobic environments and give off methane as a waste product. And so they're in the digestive tracts of cattle and deer, and actually are in decomposing materials in landfill. So methanogens we see as part of cattle digestive systems, which actually leads to not so good CO2 emissions and kind of emission stuff of carbon emissions. There we go. All right, another thing, we've got to go through it, and we'll be quick on this one because we're just going to cover some basic stuff, and we'll do more of this in class. So if you just have a few notes jotted down, we'll go through it and reorganize it together. But we will do this one because it's stuff that we need to look into. So the domain bacteria is currently divided into 5 groups. Okay, so Archaebacteria extremophiles, these are just bacteria. And it's based on some genetic sequences. So the first thing that we have is Proteobacteria. These are all gram-negative, and they share an RNA sequence. Okay? They actually have all 4 modes of nutrition, so they're going to be fairly diverse. Okay, so that's the first one. Gram-negative RNA sequences are shared. Click this button here. A good example is Thea margarita namibiensis. So it's a type of photoautotrophic species of Proteobacteria. It uses hydrogen sulfide to generate organic molecules from CO2, but it produces some sulfur waste and some small greenish kind of globules. But that's our example. So that's what you'll want to look for, is the name of the group, the general description, and an example. Okay, so that's the little green globs in it. Okay, another example is Rhizobium, and it lives in kind of root nodules of legumes, and it can convert atmospheric nitrogen to a form that their legume or bean host can use. In case you don't know what legumes are. Okay, so it's an example of symbiosis that we'll talk about later, but it's a process where life works together. So it's a close association between 2 or more organisms. Process life together. Endosymbionts is a process of life together inside, refers to one species, the endosymbiont, living within another. So that Rhizobium is an endosymbiont. Okay. Second grouping that we have is gram-positives. So these are all gram-positives. And they're also pretty diverse. So they're going to rival those Proteobacteria. They include some stuff called the actinomycetes that are common in soil, and streptomyces, which are cultured by pharmaceutical companies to make antibiotics, including streptomycin. Okay. So there's your examples. I know we're going quick through some of these, but we'll slow it down in class. The third one's called the third group Cyanobacterium. Cyanobacteria are a group of prokaryotes that are plant-like. I always think there was a particular thing on the computer, and they used to call this bluish color cyan. So it's kind of like a blue-green color. So that's how I remember it. It's blue-green bacteria, and so what's got a green color? Plants. So these guys are going to be photosynthetic. They have a particular species called Anabina, and they're able to fix nitrogen. Oops, no, I don't want to end show. Oh my goodness. I'm trying to mess up my whole thing here. There we go. I'm trying to work quickly for you guys. So here's sort of what they look like. They have those nitrogen fixing cells, and it's these series of cells kind of together. But they're kind of that bluish greenish color, so that green in there shows you it's photosynthetic. Chlamydias are the fourth, and chlamydias live inside a eukaryotic host cell, like chlamydia trachomatis, which is a common cause of blindness in developing countries, because the chlamydia that is untreated can lead to blindness. Okay, it also causes non-gonococcal urethritis, the most common sexually transmitted disease in the U.S. So chlamydias are a part of that group where there is a sexually transmitted disease, a part of that one. And that's sort of what it would look like. Sort of some cells within that. The fifth one is just spirochetes. So these are going to be our helical or spiral bacteria. There's some notorious pathogens, including syphilis and Lyme disease, which if you remind me and we get time, I'll tell you some interesting stories. We can talk about Ben Franklin and syphilis, and then we can talk about Lyme disease and my mom's Lyme disease. I think she hates it when I tell that story, and I do it every year. So get ready. I'll throw some shade on my mom. It's a funny story, though. It's really funny. We'll enjoy that one. Okay, so all organisms are almost constantly exposed to pathogenic bacteria. We can't avoid it. We can't. Most often, our body's defenses prevent the pathogens from affecting us. Go, body. Woo-hoo. Okay, but most bacteria that cause illness do so by producing poison. So there are some exotoxins. So exo meaning it sort of exits, so a toxin that exits. So this is a protein that the bacteria secretes into the environment. So secretes meaning it's pushing it out into environment. A toxin that exits. Okay, so maybe some harmful bacteria. Endotoxins are components of the outer membrane of gram-negatives that are released when the cell dies. So it's a toxin that's held within that particular cell. And so when it's kind of digested, that's when it gets released. So all endotoxins include the same general symptoms, fever, aches, and sometimes a dangerous drop in blood pressure. So Bacillus anthracis, which is anthrax that we were talking about, kind of forms these hardy endospores that have been used for biological weapons, just like I was talking about. We could look that one up a little bit. Oh, here's another one. So a weapon, the weapon form of C. botulinum, it has an exotoxin that it releases, botulinum, and it's a super deadly poison. And you know what? Like this thing is transmission and nerve signals, muscle contractions, resulting in the paralysis of muscles required for breathing. But you know, they also use it for relaxing facial muscles that cause wrinkles. So that's Botox. Is botulinum toxin A injected right into the face. So how do scientists determine which bacteria cause disease? There's a whole thing on this that we're going to talk about. We're not going to go through it. For human disease, there's this whole thing we can mention a little bit later. But it's called Koch's postulate. Oh, I didn't leave it on here. But it's Koch's postulate, and we'll talk about that one. I'm going to tell you all about this one later, Helicobacter with Barry Marshall, because that's actually a funny story, too. There's a lot of funny stories in here. So you can read through that one on here if you'd like to. It cracks me up. That's the whole reason we got it. And then he wins a Nobel Prize because, you know, he takes one for the team. We're going to talk about it. It's good. But you're going to be mad at me already because there's a 34-minute lecture. So I'm going to stop. And you're going to have learned something. And we will move forward. If you have any questions at all, shoot me an email. And we'll make sure we figure them out.